So we've made it to the nucleus, and inside the nucleus, you have chromatin. Chromatin is DNA plus histone protein with just a little bit of RNA thrown in. Usually we kind of simplify that and say it's DNA and proteins mixed together. Chromatin is also unwound DNA. It's not tightly condensed. It's essentially just organized with the proteins arranged into these units called nucleosomes. The nucleosomes are just histones packed along kind of with the DNA molecules. Now, when we do divide the nucleus, in order to essentially separate the DNA, when a cell undergoes mitosis, we will need to condense this chromatin and condense it into bar like bodies that you might have heard of before, called chromosomes. But that only occurs when a cell starts to divide. So here, what we're seeing is just DNA, that double helix with its bases in the middle, kind of like the rungs of a ladder. When you start to organize it with histones, you're creating nucleosomes. But then you can take these nucleosome structures and start to twist and twist and twist them, create a really tight helical fiber. And then twist that to create a looped structure. And then you continue to twist that, and you keep coiling and coiling until you get a supercoiled molecule called a chromosome. A chromosome is useful for moving DNA very succinctly in a kind of packaged form, but that's not useful in a cell when it's not dividing. Instead, you want the cell to have its chromatin in its nucleus a little bit more unwound so it's easier to read, because DNA contained the genetic information for how to make a protein. But during the cell cycle, we do want those chromosomes. The cell cycle is a series of changes that a cell undergoes from the time it is formed until it reproduces, and it has two periods: interphase and cell division. Interphase is kind of just when the cell grows and develops and carries on its usual activities. It does do a couple of things that are important for cell division, like in S phase or synthesis phase, it replicates its DNA. So we give double the amount of DNA so we can split it when the cell divides into two. But mitosis or cell division is really when we see a lot going on. But the cell cycle is nothing without interphase that consists of three phases, or subphases really: G1 for growth, S for growth and DNA synthesis, and G2 for growth and final preparations for division. Mitosis, on the other hand, takes up very little time compared to interphase, and it consists of four phases: prophase, metaphase, anaphase, and telophase, in that order, followed up with cytokinesis that begins kind of at the beginning of telophase, but is itself a separate process and is not a part of mitosis. And the purpose of mitosis is for growth and repair, to get more cells so a tissue or organ can grow, or to repair damaged cells. But still, most of the time, cells are not dividing. Most of the time, they're in interphase, where they have chromatin, not chromosomes in their nucleus. Their nucleus is completely intact, and at most, we might see replicated DNA and replicated centrosomes. But that doesn't last when we start cell division. When cell division begins, we kind of cease the carrying out of routine activities, and we see that chromatin start to condense. By the time we've moved into mitosis, the DNA has replicated in a process that's called semi-conservative replication. That's where you have a parent strand, and those strands separate, and each parent strand creates its new strand, and uses base pairing rules to make those new strands complementary. So the two new strands created are semi like the old strand, or half and half. This semi-conservative replication occurs during S phase, or the synthetic synthesis phase of interphase. And that's going to all occur before we get to mitosis. Mitosis is division of the nucleus, whereas cytokinesis, a separate process, is division of the cytoplasm. So at the end of cell division of mitosis and cytokinesis, we're going to have separated the DNA and the cells by separating the cytoplasm. Mitosis has those four stages: PMAT, or prophase, metaphase, anaphase, and telophase. And cytokinesis in animal cells utilizes actin filaments in order to make a cleavage furrow to split the cell's cytoplasm. But let's start first with looking at mitosis, and we'll start with prophase. Prophase is going to be the first phase where we see chromosomes start to become visible. They'll start to turn from chromatin into chromosomes. And the chromatids created will join at a central area called a centromere. And centrosomes, which are duplicated during interphase, will now start to move to opposite poles of the cell. These are going to be areas that produce the mitotic spindle. You may or may not start to see in early prophase the production of this mitotic spindle, which is made up of microtubules. But one of the key developments in prophase, besides chromosomes becoming visible, is that the nuclear envelope starts to break up. Prophase is a very long phase, so it's usually broken into early and late prophase, or in newer textbooks, it's broken into prophase and prometaphase, but we're just going to say early and late prophase. During early prophase, we start to see the nuclear envelope fragment, spindle fibers form, the centrosomes start to move to opposite sides of the cell, and we'll see chromosomes. During late prophase, we'll see the nuclear envelope almost completely gone. We'll start to see kind of the spreading out of those chromosomes, but roughly in prophase, you're going to still see the chromosomes maintain a roughly circular appearance in their location. And then we move into metaphase. And metaphase is a really quick phase, especially compared to prophase. It's when the centrosomes are able to finally line up in the middle as adjusted by the spindle fibers, and they line up kind of in the middle at something called a metaphase plate. This is an imaginary line, sometimes called the M line. And as soon as they line up, they're almost immediately torn apart. Here, where we're seeing the two parts separating, what we're seeing is sister chromatids separating. Earlier, they came together at the centromere. Now they're being pulled apart at the centromere. And we're going to see the spindle fibers pull them towards opposite sides of the cell. While metaphase is short, anaphase is even shorter. Almost as soon as the chromosomes stop moving to opposite sides of the cell, we have telophase, which will begin separating the cells into two. It's basically the complete opposite of prophase. So we're going to see chromatin return, the spindle fibers disappear, the nuclear membrane come back. And in telophase, cytokinesis begins. It sometimes begins during late anaphase, but definitely has started by telophase. And we're going to use a ring of actin microfilaments to kind of go around the middle of these separating cells, kind of like a lasso, to create a cleavage furrow as it pulls tight. That will create two daughter cells that are identical to their parent cells, because mitosis is about not just replicating DNA and separating cytoplasm, but creating clones in order to replace or grow tissues and organs. So here is telophase and cytokinesis. The biggest difference between telophase and anaphase is you actually start to see cytokinesis, or the separation of the cell cytoplasm during a telophase, whereas we don't really see that cleavage furrow formation during anaphase. So what we were seeing as we talked about not only DNA and whether it's chromatin or chromosomes and looked at mitosis is this process of separating the DNA and its replication. But why are we so concerned with DNA? Well, that's because it's the genetic blueprint for how to make a protein. And there's this idea called the central dogma of molecular biology, wherein DNA has the instructions, RNA is the go-between molecule, and it's going to help us make protein. DNA is also stuck in the nucleus, and we need RNA, which can go to the nucleus and the cytoplasm, to get the instructions to a ribosome so we can make a protein. Protein synthesis uses DNA as the blueprint, but not all of DNA. While it's the master blueprint, we use just parts of DNA, which is a gene. And we actually read DNA bases in threes, which is called a DNA triplet, and this helps us kind of discern or read the genetic library. Remember that DNA has bases that include A, G, T, and C, or adenine, guanine, thymine, and cytosine. And each triplet, in a specific order, is going to code for a particular protein by coding triplet by amino acid. So when we do protein synthesis, everything we make from reading DNA is not necessarily important. Some of the pieces need to be cut away. Exons are going to be important pieces of the genetic code, that code for specific amino acids, and introns are going to be non-coding segments. So RNA and a complex called a spliceosome helps kind of essentially make the RNA we want that looks exactly like what we need, that only codes for amino acids. It's basically going to be a DNA decoding mechanism and a messenger. There's three types of RNA. Messenger RNA, which is the messenger from the nucleus to the cytoplasm. Ribosomal RNA, which makes up part of a ribosome and is made in the nucleolus. And transfer RNA, which brings the right amino acid to the ribosome to make a protein. It helps essentially assemble what the messenger RNA is bringing to the ribosome. And keep in mind that RNA does differ slightly from DNA. Not only is DNA double-stranded and RNA is single-stranded, and they're found in different locations, but RNA has the base uracil instead of thymine. So RNA is our go-between molecule. It links DNA to proteins. It's essential in the central dogma of molecular biology. And all RNA is formed in the nucleus. And again, don't forget the three types of RNA and what they do. Messenger RNA, or mRNA, carries instructions for the building of a polypeptide from the gene or segment of DNA in the nucleus to the ribosome in the cytoplasm. Ribosomal RNA, or rRNA, is a structural component of ribosomes that, along with the tRNA, helps translate messages from mRNA. And transfer RNA, or tRNA, binds to amino acids and helps to pair them with the bases of codons that are on mRNA. Just as we read DNA triplets, we read mRNA in threes called codons, and those are what are read at the ribosome to begin the process of protein synthesis. And protein synthesis occurs in two steps: transcription and translation. Transcription transcribes DNA into RNA, and that's a process that keeps the molecular information pretty similar. DNA and RNA are both nucleic acids. Translation turns RNA, specifically mRNA, into a protein or polypeptide. So that's changing the molecular language. Transcription occurs in the nucleus, whereas translation occurs in the cytoplasm, but specifically at the ribosome. And transcription is going to create a kind of complementary base sequence, but of RNA instead of DNA. And there's many different transcription factors and gene activators and promoters and this entire complex that helps facilitate this process. Enzymes involved include RNA polymerase that oversees the synthesis of mRNA and unwinds the DNA template. And there are both start and stop spaces for transcription factors and all of the complexes that help this process take place. Translation is also able not only to convert base sequences of nucleic acids into the amino acid sequence, but utilizes both mRNA, tRNA, and rRNA. And we actually can really kind of review how this genetic code is read. This graph isn't rendering correctly, but it's essentially a 3-base sequence on DNA that represents a codon on mRNA. Then we take that mRNA codon, like UUU, and we match it up using a codon chart. And a codon chart will dictate to us which amino acid matches that mRNA codon. In that case of the example I gave, UUU, in the upper left-hand corner of this image, it would be PHE, or phenylalanine. There are also start and stop codons that allow mRNA to know where to start and where to stop. In order for translation to occur, the mRNA attaches to the small ribosomal subunit, and the large one comes in, and we start with mRNA, and tRNA brings its amino acid based on an anticodon that matches the codon. So there's kind of a puzzle piece formation that occurs during translation to match mRNA codons to tRNA anticodons, and making sure we put the new amino acids in the right order. And this will continue until a stop codon is reached on mRNA. And that's when the entire ribosomal complex kind of falls apart, because we have what we need. We have the protein. During this entire process, complementary base pairing directs the transfer of genetic information in DNA to amino acid sequence. So we go DNA triplets to mRNA codons, and that's transcription, and mRNA codons to amino acid sequence, or protein, is translation. It's triplets in DNA, codons in mRNA, and anticodons in tRNA. And anticodons pair with codons. And we still use base pairing rules, but keep in mind base pairing rules are slightly different in RNA. A or adenine pairs with uracil in RNA, and A or adenine pairs with T or thymine in DNA. So that's kind of a summary of how we're getting from DNA to proteins. But there are other roles of DNA. DNA codes for other types of RNA, like microRNA, which can bind to and silence mRNAs made by certain exons, or riboswitches, which are folded RNAs that act as switches that can turn protein synthesis on or off in response to certain environmental conditions. And even siRNAs, or small interfering RNAs, which are similar to microRNAs, but they can actually be made to silence mRNA from a pathogenic source, such as a virus. So these are just other ways that parts of the cell can modulate itself and its functioning. So as far as it goes for chapter 3, that's the end. But make sure you go back and you review things, especially like the organelles, the steps of mitosis, and the general rules for making DNA, both semi-conservatively, and then turning DNA from a triplet into codons and then later into protein sequences. So those are all things I would look back on.